Now we're going to start on part 2 of the history of microbiology by Dr. Roderick L. Roll. So I drew a concept map. We have our trunk and our 3 branches. This slide is showing that we're going to talk about the early years of microbiology, the golden age of microbiology, and the modern age of microbiology. How can microbes be classified? So in the early years, Carlos Linnaeus saw many organisms. He saw plants and he saw animals. And he saw that some of these organisms had similar characteristics and some of them did not. So Mr. Linnaeus devised a taxonomic approach. He devised a method to group these organisms into categories based on similarities. So he is the father of the taxonomic system, Mr. Linnaeus. Name the microbes of Leeuwenhoek. Those microbes that Leeuwenhoek discovered 100 years ago are going to be called class chaos. So you'll see that Linnaeus didn't really know what to do with those microbes that Leeuwenhoek discovered. So he just put them into a class called class chaos. By the end of the 19th century, chaos were called microorganisms. So class chaos had bacteria, had fungi, had protozoans. All of these which were discovered by Leeuwenhoek put into a category called class chaos by Linnaeus. So by the end of the 19th century, about 200 years after Leeuwenhoek's discovery, they're going to be commonly called microorganisms. Leeuwenhoek's microorganisms were grouped into 6 categories: fungi, protozoan, algae, bacteria, archaea, and small animals. This slide is just talking about the basic cell types. We have eukaryotes versus prokaryotes. This slide is basically saying that, all right, yes, Linnaeus came up with a taxonomic approach. Later on, scientists realized that we're going to, since we're talking about grouping systems, we're going to have a grouping system based on if the organisms have a nucleus and if they do not have a nucleus. Now, this is much after Linnaeus, because one thing is evident in this slide: some things have to be looked at with a microscope, a very powerful microscope, to see if the organisms have a nucleus or not. So this is called super kingdoms. So there's 2 super kingdoms, prokaryotes and eukaryotes. Eukaryotes have a true nucleus. You can see the nucleus, and we'll talk about what a nucleus does later. But organisms that would fall under this category would be plants, animals, fungi, and protozoa. So all 4 of these organisms or organism categories would have a nucleus. This category is prokaryotes. It does not have a nucleus. So microbes are going to fall under this category. Now, not the microorganisms of Leeuwenhoek, the microbes of the 19th century, the 20th century, the 21st century. Now, when we talk about microbes, we know that we're talking about bacteria. All right. So this slide is showing us two major scientists, Linnaeus. Linnaeus proposed only two kingdoms. All right. He proposed that everything that he saw and analyzed fell into two categories: Animalia, one kingdom, and Plantae. So this was in the 18th century, about 100 years after Leeuwenhoek started talking about class chaos and his different microbes. Later on in the 20th century, about 200 years later, Whitaker said, no, 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 no. There's not only two kingdoms, there's five kingdoms. So Whitaker proposed a widely accepted taxonomic approach based on five kingdoms in 1969. His five kingdoms were Animalia, Plantae, Fungi, Protista, and Monera. So understand that in the 18th century, they had very powerful microscopes of that day. But in the 20th century, the microscopes were even greater. You can pierce into the cells. You can look into the nucleus. So because of that, they had to regroup everything. All right. So Linnaeus is the father of the taxonomic system, but his system has been modified several times since it was invented. So what is taxonomy? What is taxonomy? All right, there's three bullet points under taxonomy. So this is what taxonomy is. It is a classification scheme. All right, it is a nomenclature scheme. It is a identification scheme. So classification is nothing more than grouping. I'm going to group all of these organisms. If I had a container full of shapes, everything that had a square shape, I would put it in one group. Everything that had a triangle shape, I would put it in a different group. So this would be a classification scheme. All right. Well, how about the actual shape itself? I named it a square. I named it a triangle. Okay. The naming of it would be nomenclature. All right. So the naming of it would be the nomenclature and identification of it. I got to see it. I got to use visual mechanism to see the square and see the triangle and see the circle. So this is what taxonomy is. I'm going to group everything that I see. I'm going to name everything I see. And I have to see it. I have to be able to identify it. I have to be able to see it. I'm going to jot down a description of it. And that is called identification. So all three of these bullet points fall under taxonomy. Taxonomy enables scientists to organize large amounts of information about organisms and make predictions based on knowledge of similar organisms. All right, so this is what taxonomy is. So the first one we'll dive into a little further is classification. Grouping. I'm going to put everything into groups. Here's the grouping system. You start off with the most inclusive. The most inclusive is up here, followed by the least inclusive just down here. So this one up here would include a lot of organisms versus this one would only include one organism. So kingdom, phylum, class, order, family, genus, species. If you took a course like Biology 2, you would have to learn all of these different groups for specific organisms. So for example, Homo sapien. Homo sapien would be down here. That's the least inclusive. That's very specific, but it would belong to kingdom Animalia. Kingdom Animalia have things like sharks. It has things like monkeys. It has things like squirrels. So it has all type of things under kingdom Animalia. All right, so just understand that this is the way that Linnaeus grouped everything. So there was a way that we remembered in high school. King Philip came over for good sex. So that's how we remembered it. So until the 1960s, kingdom was the highest classification category. Now we know that they have super kingdoms. So the super kingdom is prokaryota and eukaryota. This slide is showing us that here is a classification scheme based on domains. So we're going to talk about who discovered domains shortly. But there's three domains. Bacteria is a domain, so all the microbes that are bacteria would fall under that. Archaea is another domain. All of the archaea, like extremophiles, would fall under those categories of domain. And then eukaryote. Eukaryotes are things like animalia, fungi, plants, and so on. So this is another classification scheme, but it uses something that's way above kingdoms. It's called a domain. Now, organisms' genetic material led to the proposal of the domain system. And like I said, we'll discuss this genetic material as being RNA a little later in our talks. So we're looking at the three domains. Carl Woese compared nucleotide sequences of ribosomal RNA that were collected from different organisms. He saw that the rRNA rarely changed. And because of that, he was able to group all organisms into three domains. The three domains are eukarya, bacteria, and archaea. The three domains were determined by the ribosomal nucleotide sequences. All right, so this was a much later taxonomic approach that was a little different from Linnaeus and Whitaker. Linnaeus only had the two kingdoms, Animalia and Plantae. Whitaker had the five kingdoms, but they were still looking at physical characteristics in most of these. Now we're actually looking at molecular characteristics, rRNA. This is a combination of all three of those, Whitaker, Linnaeus, and Woese. Look at how this concept map flows, or this branch diagram flows. We have the domains up top. We have the domains. We have the three of them. We have bacteria, archaea, and eukarya. Let's just follow the eukarya. We have different kingdoms. We have Animalia, we have Plantae, and we have fungi. The Animalia has different phyla. The phyla Chordata, Arthropoda, and so on. We're going to just follow the Arthropoda on down. So Arthropoda has different classes within it. It has Insecta, Crustacea, and Arachnida. So these classes would fall under here. So do you see how the classification scheme flows? All right. Now from class, we have King Philip came over. So the order would be next after Arachnida. So we have our order, followed on down to our family, follow it down to our genus, and then we finally have our species. All right, so this is our classification scheme. So based on Linnaeus, we're looking at taxonomy. Taxonomy has a major goal. So the major goal of modern taxonomy is to reflect their deviation from common ancestors. Remember, taxonomy had three subcategories under it. So the first subcategory of taxonomy is, as we stated earlier, classification. Classification is also referred to as grouping. All right. Grouping used to be based solely on physical and chemical characteristics, but now we use molecular characteristics. We use things such as ribosomal RNA to group organisms. The second subcategory is nomenclature. Linnaeus provided a system that standardized naming of organisms based on characteristics they have in common. He grouped similar organisms that can successfully interbreed into a category that he called species. So, Homo sapien is a species. So, all organisms that can interbreed would be under that same category. Blacks, whites, and Asians, or Mongoloids, Caucasoids, and Negroids. So all three of those groups can interbreed. So they are called a species. Binomial nomenclature is given two names for every organism. Every organism has to have two names, binomial, two names. The two names are going to be a genus and a species. The genus is the noun, and the species is a specific epithet. It is usually an adjective. So, Homo sapien. You can never write just sapien. If you just write the sapien, it's wrong. All right? So it has to have two names. That's why they call it binomial nomenclature, two names. Every organism on the planet has two names. When you're writing it, it needs to be written as italics if you're typing it. If you're writing it, you have to underline it. Homo sapien. Make sure you do that. And all taxa are Latinized. And finally, the third subcategory of taxonomy is identification. You have to be able to identify the organism.